Well, hey again, Mr. Bleeker here. This is our fourth lesson in the DNA series entitled Mutation. We'll be looking at mutation in a couple of different ways. Mutation caused by radiation damage. We'll look at that. We'll also look at mutation as a process of sort of recording errors when DNA is replicated, and these little errors that pop up that change the way that genes are expressed. If you recall from my Biology 11 series, mutation itself changes the genetic code at a very low lying level to the point where we start to get some changes in the overall genetic code. While sexual transmission of genes may do the swapping of the cards, mutation creates new cards. And I wanted to stress that point. Mutation can be very detrimental. Well over 99% of them result in non viable organisms. But for the changes that turn out to be advantages, well, you'll see in this lecture series, some can actually improve survival rates. This is how we get new versions of things. It's basically through trial error and that good old natural selection. But I digress. We'll get going. Okay, so there we go. Awesome. Push over the thumbnail tray. So the reason I put corn on this particular slide is because corn itself is a genetically modified organism. If you've eaten corn that you get, for example, from most stores, you'll sort of see a uniform sort of white corn or yellow corn. And that's rather odd when you think about it because each of these kernels here, is a separate organism. Each one can replicate the corn plant. Each one has its own genetic complement. So you can see what's called variation. Now, to be specific, a variation in their phenotype, the physical manifestation of their characteristics. They've all got different genes. So when you look at what they call Indian corn or this particular version of maize, you should see variation. A good question is why you don't. Well, the reason you don't see a lot of that variation is because it's been genetically modified so that corn appears in a uniform way. And that was thought to be more attractive to consumers. This is more ornamental, but this is actually the corn of tradition. It should look different. They should all be kind of unique individuals, each little kernel there. But when you look at what you're consuming today, it's genetically modified. So if we talk about genetics, we're really looking at the transmission of genes, genetic codes, from one generation to another. And when you look at the whole purpose of sex, it's to come up with new sort of shufflings of the deck, right? To give the next generation a little bit, to shuffle the next generation, to see if that generation can survive better on the planet. When you look at germs, if you think back to Biology 11, when they have sex, they're trying to come up with new organisms that can stay ahead of the antibiotics that we throw at them. The same thing's going on with malaria, for example. It's a very resistant organism, becoming resistant to the drugs that we throw at it. But when you look at mutation, as I said, that those sort of new cards in the genetic deck, if you shuffle these, then the population starts to get new characteristics over time. So as far as that evolution goes, we can see the evolution of characteristics, but that starts at the genetic level, at chromosomes and genes. So, said pretty much all I want to say here. So to get these new cards in the deck, there's sort of natural low-lying mutation. If you think back to good old Mr. DNA polymerase that we were looking at, DNA polymerase has a built-in spell checker, and it will fix genetic errors when it copies the DNA molecule. However, some of those genetic errors aren't fixed. Sometimes the sequence of the daughter strands is different than the parent strands. So semi-conservative replication isn't perfect. And you would think that you'd want it to be 100% perfect, but if no new cards can be inserted in the deck, you wouldn't get any new games. Let's put it that way. So you want mutation so that you get some gene variants And then when DNA transfer occurs, you're transferring new information throughout the population. This is always going on at a very low level. Mutation is the stuff of evolution. It's really one of the basic mechanisms. Whether or not those mutations are an advantage, that's different. I always like to say if you had a mutation, if you were a rabbit in the middle of winter and you had a mutation that caused your fur, instead of going white, to be a brilliant bubblegum pink, you probably wouldn't survive. That would be a terrible mutation. Albinism is a mutation you'll see in animals that really doesn't improve survival rates. That's why a lot of these organisms get picked off, and that mutation results in non-viable organisms that just tend to die out in populations. That's if there's enough pressure against them to die out, per se. So mutation is something that's inheritable. You can get a new DNA sequence, and that can show up in the germ cells, the egg and sperm of organisms. Now, it does occur when you're getting some DNA replication with errors, and you can also get some changes to genetic expression due to viruses. Viruses can insert into your genetic code, and they can, when they put their information in, it can cause some variation. But mutation, as I say, DNA polymerase making errors is pretty common. Now, this is where I've been wanting to get to. What kind of mutations can happen? If there's going to be a mistake by DNA polymerase, what is that mistake going to look like? This is where I want to go out for a second. So I'll talk about what they are, and then we'll look at them. If there's a mistake with DNA replication, and I'll just use yellow here. If in a DNA strand, we'll just put in some sample nucleotides here. Off the top of my head, there we go. If it turns out that you get a substitution, if one of these was changed, just a copying error, just sort of DNA polymerase messes up. If there was a change of one of these to something different, what would happen is when that DNA would get transcribed, now you would have a different transcription. What we would have expected to get originally, and I'll just choose, let's choose an opposing color. We'll start with orange. You would have initially expected on mRNA to get U, G, and I think that was originally a C. There we go, UGG, and CGU. But due to a mistake like this, what you'll get, so we'll just grab the eraser tool here for 1 second. There we go. You won't get this, which is what you would have expected. Due to that mutation, instead what you'll get, there we go, this being G, you will get a new insertion. There you go. I'm going to go back to the pencil tool. There we go. You'll get this one. And that's not what was anticipated. And it really does affect things. It will affect, of course, the first mRNA codon. Now, I'm just going into one here, but this is if you get a substitution. So we'll look at the net results of what could happen here. I mean, it is possible you could get a stop codon, which would just be no expression at all. That would be a disaster. It's not just base substitutions that you can get. Sometimes one of these can literally be eliminated. And if you eliminated one of these, that could be a major problem. Let's just undo what we did there. Get rid of some of this. There we go. You could alternatively put in another one. So if you inserted an extra A here, for example, now you've created some interesting problems with the reading frame. And that's what we're talking about down here. Inserting or deleting portions of the genetic code. As a summary, I'd say the base substitutions tend to be the most benign, but insertions or taking things out, that changes the whole thing. Because when you read the DNA alphabet, there's no spaces between the words. It's like reading a book where there's no spaces literally between anything. And it can really change the reading frame of the sentences. So let me show you what happens when each of these inevitabilities occurs in nature. Okay. Okay. So we'll go to the first possibility here. When your DNA, here we go, when your DNA is being copied, remember this is going on all throughout cells, and you've got all this chromatin and DNA molecule being looked at, there could be little errors going on all over the place. Now, what might be some things that could cause mutation? Well, copying errors by DNA polymerase could be caused by things in the environment. It could be radiation, right? Think about skin cancer, for example. That's a mutation of DNA that can cause skin cancers to go melanomic, right? You could also get toxins in the environment. PCBs, organisms come into contact with things that poison them, and DNA polymerase doesn't do a great job copying the DNA molecule when the organism's being poisoned. Smoking, when you look at the development of lung cancer, right? The development of liver cancers, these things that change the cell or turn on genes that should not be on. Now, most of these mutations are just handled, but some stick. So let me just move forward here a little bit. There we go. Okay. So the two types we'll be looking at are substitution mutations of just like one of the genetic bases, and then we'll look at taking out bases or adding them. It's the second one that makes a really big change. Okay, so normally, if we were looking at a DNA coding strand, if this is what we expected to see, this should be the mRNA. Now, if you read the codon table, UUU codes for phenylalanine, UAU tyrosine, GCA alanine, and CGU arginine. Sometimes pronounced arginine. I always get that wrong. You get the idea. Now, there can be what we call point mutations. So in this case, I'll just play this out a little bit here. A silent mutation would be one, and we'll just come up on the marker here, where one of the bases has changed. Now, what we would expect is this single change, you're hoping that it doesn't have an effect. So what if that change happens, but you get lucky and you still get phenylalanine, which is expected as a result? Well, then there's no change in the protein. It's a different codon, but you got lucky. You still got phenylalanine there. So that's good. So we call that a mutation, but it's silent because it doesn't affect the polypeptide as a result. Okay, so I call that one silent mutation, the you got lucky mutation. Now you can get something which is called missense, where a mutation happens, and now you've got a crazy word in your DNA sentence. So if a change happens, we'll go down here a little further and change this cytosine, or sorry, this guanine, a little bit different. You originally expected to have, let's go to alanine, a G, C, and an A. But now what you've got is you've got GGA. So that's the point mutation that's as a result of this alteration here. So you've got a different codon because of the DNA mutation here. Okay, let's be clear about that. It was the DNA mutation. So you're getting GGA. So you go read a three-way table and you say, I was supposed to get alanine here, but now I've got glycine. And that's a problem because in some cases, a single amino acid change can cause a change in the overall protein. It's like you're building a house and you're supposed to put in bricks of certain sizes, and then you come along and you put in one part. Now, you got to remember, proteins are really foldable things, and this has caused major issues in nature. I'm going to give you a perfect one. It's a classic stock case in point. Here's the DNA sequence that produces normal red blood cells. So the amino acid sequence for their sort of their structure here is laid out here, and at the bottom, what you'll see is the complement, sort of more or less to that sequence. So we're looking at the double-stranded DNA here. Now this calls for glutamic acid. So normally the way the structural protein that holds up red blood cells and makes it so that they are sort of these hollow discs like this, or biconcave discs, you would expect it to be valine and so on. And glutamic acid really needs to be the support structure in this corner to hold up, to make red blood cells hold up the way they do. Now there's a point mutation that has happened. It was a DNA copying error, at least that's what research indicates. And instead of this DNA sequence here on the strands, instead what we're getting is we're getting a mutation. So adenine and thymine are in the strand. Now you might say, well, which one mutated, the adenine or the thymine? It really depends. If this side had mistakenly put in a thymine, when it's copied, you would have gotten the adenine. If this side had put in the adenine first, well, if DNA copying happened regularly, you'd get thymine on this side. So these would be the matching strands. Here's the point. Everything is normal right up to the point of this missense mutation, and you get here, and the mRNA complement to that, you don't get glutamic acid, now you get valine. Ooh, that's a big structural difference. If you look at sickled red blood cells, they're not like this all the time. What will happen is under times of stress, you could think of it as a structure that if you put too much pressure on it, it will just sort of go this sickle shape or this crescent shape. Now you say, well, why, is this a fatal problem? It's kind of a two-part explanation. When you look at regular red blood cells, they're this biconcave shape, and they fit really nicely through your blood vessels, right? Think about your capillaries. They're really thin-walled, right? They're like one cell layer thick. So these are quite flexible, and they can sneak through your capillaries from your arterioles to your venules, get around your body, so to speak. That's good. Now, when you look at sickle cells, if you've got sickle cells which are expressing that mutation, they can clump up, and this becomes a problem. I've got a friend who suffers from sickle cell anemia, and when she's stressed out, what can happen is her red blood cells will be like this most of the time. She gets stressed out or suffering from an infection, and it's like her red blood cells suddenly go crescent shaped. It's reversible is what I'm trying to tell you. And when this is happening, she's not getting good blood flow, and she's just flat on her feet. She's lying down. She's not getting properly oxygenated and totally tired. So this becomes a problem, reduces blood flow. It's not really a blood clot. It's not good, but it doesn't have to be fatal. Now, you're thinking, well, why could this possibly be a good thing? Like, you see sickle cell anemia in a lot of African heritage. You'll see it more in people with darker skin. And you wonder, well, why is that happening with people of African descent in particular? Well, if you think about malaria, right, the Anopheles mosquito is a major problem when you go to a lot of countries that are closer to the equator. And in Africa, this is a major problem. In grade 11, I talked about sleeping under mosquito netting, wearing bug spray, doing anything to keep these guys from biting you, because if they infect your liver, they'll get out of your liver, they'll get into your blood, and they'll pop your blood cells, and you get into this horrible fever chills problem. And of course, the cycle repeats when somebody who's got malarial infected blood gets chomped on by a mosquito, and the mosquito now is one of the hosts, and the malarial parasite goes through a reproductive stage in the mosquito gut lining until it burrows its way up to the salivary glands. And once the salivary glands are infected, the mosquito is basically a hypodermic needle to transfer the sporozoites, which is the one of the generations, into your body. Okay, so here you go. So malaria, the vicious cycle. It turns out that this mutation that we're looking at here is a good defense against malaria. Well, why is that? Because this is very hard to infect. The malaria parasite can't really get in there. So this turned out to be a defense mechanism by accident that showed up in the population. So so a mutation led to a good thing and increased survival rates. So as when someone was being infected like this, those blood cells under stress would do this, so that the malaria parasite really couldn't do a good job of infecting the host. So it led to greater survival rates in the population. And so this mutation just kept getting passed through regular sexual transmission like a survival card, and it stuck. That mutation had a reason for its existence. A bad thing turns into a little bit of a silver lining, if you will. So that's an interesting sort of case in point as far as what we were looking at goes, as far as a silent mutation happens. Not bad. Not bad. Now, there's a little bit more to this. Let's just play on. If you got, a missense mutation. Silent mutations cause no changes. A missense mutation. Now, moving on, clearly. A nonsense mutation would be, let's say we had a change there. So it's just a single point change, just a base substitute change. And let's say you get UA. It's like, whoa, wait a minute. What does that mean? This here, if you look at your three-way table, this is not going to be some different amino acid. This is one of the sequences which calls for an all stop on protein production. Ooh, that's not good. Because if this were a protein, let's say that you needed to survive, let's say that was the production of insulin or something. Now you're not getting the production of insulin anymore. Well, that could just very well be a fatal mutation. Hopefully, this isn't a protein in the body that's so important that you couldn't live without it. That's probably the worst case scenario, is to get a nonsense base mutation in that point. Okay, now frame shift mutations can be quite a bit more serious. Just pause this for a second. If you notice the yellow here, you'll notice that these amino acids are all different from the point where the mutation happened onwards. And frame shift mutations are classic for that. If you change the reading frame by either putting in a base, or rather a nucleotide, or if you take away a nucleotide, you're going to shift the reading frame. So the codons are going to get read 3 at a time, but now it's a different reading frame. So let's look at that in a little bit more detail. Okay. So at this point, we're going to insert a thymine into the DNA sequence. So in turn, when the mRNA is grown in, right, RNA polymerase comes in, now what we have is we have a little extra match here. Okay, so our codon reading begins this way. So we've shifted a little bit to the left, right, because we've inserted a new one here. It's like, oh boy. Okay, so now instead of reading the way we would have expected to here and then moving on to here, now we're reading from here. The reading frame has gone that way. So sometimes these are called shift lefts or left shift. And as a result, you get, let's pause that. Went a little quick. I just want to move back. A little bit more. Come on back here. There we go. As a result, let the amino acids come back here. Oh, look at that. Okay. Well, I guess it's just as nice twice. Okay. So we expected this to produce our phenylalanine, but now as we read downstream, AUA gives you this amino acid, this gives you this amino acid, and you get the final amino acid. They're not what you expected. It should have been tyrosine, alanine, and arginine. Your protein is radically different at this point. That's a big change. If your protein's expected to do a specific job, maybe this is becoming part of an enzyme, this is not going to be the same product you had before. So, big issue. Now, the final one would be what would happen if you deleted a nucleotide at this position. Okay, so now you'll see your reading frame will sort of back up here a little bit. So what does this result in? There we go. If you look at the matching mRNA sequence, you get UUG, CAC, and it should have been, right, there should be, in fact, we expected to have a final nucleotide here, but the next one from the right is going to have to move over. Okay, so we've got a frame shift, which is a deletion. So let's see what the result of that is. Okay, so this is very different. We expected tyrosine, alanine, and arginine after. Now we're getting leucine, histine, and valine. Okay, so again, that's a dramatic shift from the amino acids that we expected to get. So what does this mean? This means that if you add in a base, a nucleotide, you can create a shift which will affect all the amino acids downstream. Alternatively, if you take away an amino acid, it does the same thing. It's just which way the reading frame shifts. And these are all like major mutations that have to be considered. Okay, good little animation. I'm going to go back to just our lecture notes for a moment here. And my dog's wanting me to hurry up and finish this lecture. You can't see it, but as I'm doing this, she's constantly pawing at me. That's okay. Jenny will get a walk soon enough. Okay, I like to do the mutation thing here. When you look at Magneto from the X-Men, there's this Hollywood genre sort of this idea that these mutate. There's a mutant gene and it gives people powers to manipulate magnetic fields, and oh my goodness. And the reason I put that in there is mutation's not like that. It doesn't change the whole organism. You know, it's not like that. I think this is sci-fi going a little bit crazy with the idea of mutation, right? To the point where we have a whole new suddenly just through this new gene that we've got a whole new species of human. I think they call the mutants in Marvel Comics, they're Homo superior. Nice name, hey? Now that we've looked at the different kinds of mutations, missense where an incorrect amino acid may go into place, or nonsense where production stops, and the third possibility is silent where you get lucky, and you get the same amino acid. Well, I mean, these are really the mutations that are going to happen. They're going to make small changes in organisms, unless that mutation happened in something so critical that the organism doesn't even develop in the first place. I mean, that is possible. Now, alternatively, if you were looking for big changes, it's not the point mutations where odds are you'd see the big changes. The big changes you would expect to see would be in the group that's coming up, of course, which is oh, it's coming. I've already gone over this, but I'll pause. This is just the bit on hemoglobin, where you get sickle cell anemia as a result. The valine instead being the sort of the mutation in all of this. Just pause, give you a chance to look that over. Pet my dog. If dogs could talk, she'd be asking for a walk right now. In fact, she is. But the insertion mutations, as I say, are the ones where you would expect to see the big, big changes. For example, one like this, with the insertion of a nucleotide at that point. That's huge because you should be seeing this repeated amino acid sequence in this protein, and now you've got this down here. That may not be a good thing for the organism. And the extra nucleotides make it a little bit crowded in the DNA, right? If you're going to insert them, that's just DNA polymerase getting distracted and putting in an extra nucleotide or a couple of nucleotides as it's going along. Maybe it got distracted by the UV light that was bombarding it as you were sitting out in the sun. UV light is very energetic. There's reasons why when we go to the doctor, we wear a lead apron when they take x-rays because DNA polymerase is doing its thing all the time. And high intensity radiation, well, it would be like trying to study but being hit with constant bombardments of particles. It could be a little bit distracting, right? Too much energy, too hard to do your job while you're getting sort of whacked continually by high energy radiation in the form of UV light or gamma radiation. Heaven forbid for that. So frame shift mutations, as we say, they can cause really big frame shifts. And these can be problems. They call these knockout mutations because if you get a really big change downstream from the initial codon, it can result in fatality. The whole fatality of that gene expression. If, like I say, if that gene was for insulin and you had a mutation, you can pretty well forget it. It's not going to work the way it's supposed to. You've changed what the resulting blueprint for that protein, be it an enzyme or whatever, will look like. Later on, we'll talk about transposons or jumping genes. Kind of interesting. We do have some genes that can move around in our DNA, and if they move from one section of the DNA to another, they can sometimes interrupt the DNA sequence. And that can be a little bit of a problem because they can cause these insertion errors. And you've seen it change the reading frame. Transposons are really common in corn. And in corn, we can get a lot of variation as these genes jump around. You tend to think genes are just sort of like static elements that stay in just one spot. But that's not true. There are some that jump around, and they will cause these insertion mutations. And that's what you're seeing here. So when you look at this variety of maize, sure, they're all genetically different and they should come up differently. But even within each of these kernels, within its genetic complement, it's an interesting observation that they couldn't really explain how we were getting variation when there was asexual reproduction going on. It's like, what's going on? There's no new genes here. Nothing new is being introduced, but we're getting variation within the organism, really, even when it isn't reproducing. So we were getting some color variation that was showing up. And some of it was explained by these transposable elements, where there's the non-pigmented, partially, and normal pigmented corn. And as that gene would land in different positions, you get different protein expression. So you got reds and blacks and things like that, right? So pretty neat work by Barbara McClintock in the 1940s. It took a long time to be recognized for this, by the way. They didn't really understand what was going on, why you were getting this variation, even when there wasn't sexual reproduction going on. So none of these plants were bioengineered, but looked like they were just spontaneously mutating right in each kernel. So neat little bit of history here. When it comes to mutations, the mutations that occur spontaneously, they're kind of rare going on, but mutagens can really speed up the process. If you look at mutations, I mean, in the tree of life, we've got all these different organisms due to environmental selection acting on the DNA that's there. And the DNA is changed due to mutation, right? So there's been a selection process. If you had really bad genes, you were gone. And if you had genes that were really quite good in your environment, hence the long necked giraffe, hey, that was probably a good gene, improved your survivability rate. That gene became really common in the population. But certain chemicals and radiation, as we said, they're mutagens. And smoking, for example, increases the prevalence of lung cancer. So you can really mess with DNA polymerase when it's replicating, and something that does is called a mutagen. Ultraviolet radiation can be pretty nasty for this. Gamma rays, as I've said, X-rays, and these can cause some pretty nasty either substitution mutations or deletion mutations, insertion mutations. Not good. But generally, mutations tend to be rare. DNA repairs itself. Like, it's not like DNA polymerase doesn't come by again and again, right? And the molecule's constantly being examined, replicated. And so we've got genetic defense mechanisms to sort of safeguard against these, but you got to realize those mutations can slip past every now and then, right? And there are things, the mutagens can mess with the entire process. So DNA polymerase's spell check is really reliable, and it's why we still have vaccines against polio. That's a DNA virus, and because when its DNA is replicated, it stays the same all the time. Well, that's good because we have a really well working vaccine against that stable version of the virus. RNA, well, that's different. When you look at the cold and flu viruses, they don't have a DNA spell checker. If you choose RNA as your genetic code, no spell checker. It's going to be a lot of mutations. So that proofreading is good. Usually mismatch repair can happen. Right? DNA can go through there and fix things up. But there's some little joke here at the end. You're lucky nobody was injured. Frames are out of repair. We don't generally have to worry about this. Now, just show you some of the ways. Just have one more tidbit and I'm done here. In the interest of time, show you why UV light or UV sterilizers in your hot tub, for example, or UV. I have this in our furnace where when the light goes by, there's a big old UV light. Well, if any germs are floating through the air, UV light is an excellent sterilizer. Let me show you what UV light does. On one DNA strand, if there's a couple of thymines, it happens with thymine in particular, UV light can supply just enough energy to cause two thymines on the same side of the tracks to get glued together. Why does this matter, you might ask? Because you can't replicate these guys afterwards. In other words, they get so glued that replication of the molecule after this point becomes a serious problem. The DNA is more or less stuck. Now, this is good if you're trying to kill germs, because if germs can't replicate their DNA, they die. It's a sterilizer. Think of it this way. UV light hits the molecule, and you form what's called a dimer between two adjacent thymines. Now, good luck replicating this molecule. It sort of takes it out of the equation. It's sort of checkmate as far as this DNA goes. Here's another view of it, a thymine dimer, and it's disrupted the hydrogen bonds between the adenines and the thymines because the thymines are suddenly really interested in each other. Well, that energy was supplied by UV light. When I was at UVic, we used to turn on the UV floodlights in the microbiology labs just to do this to any germs that might have accidentally been left behind. Why do that? You can't replicate this molecule now. And if the organism can't replicate its molecule effectively, it dies. So UV light is a tremendous asset in purifying water, in purifying air. So you got your incoming photon, and here's what you're starting to see. Some of these dimers begin to form, and it makes the molecule just non-replicable, and that's good. Gets rid of germs and things like that. Same thing with viruses. If they're hit with intense UV light like this, they can't replicate. That goes for the single-stranded viruses. So I guess if we wanted to keep healthy, we'd just get everybody out of the school and turn on the UV lights, right? Sterilize everything. But you got to remember, that's only one part of the equation, because while you could do this in a hot tub as the water is circulating around and you're killing germs, that you have to get the light to like every surface. So you still are going to need your strong chemicals. Remember how well bleach worked, right? When we did our test of sterilizing agents. Yeah, it's sort of all hands on deck in keeping germs under control. So, anyways, that's it. That is our section on mutation. There's a little bit more. It's a very large chapter. I find it a very rewarding chapter. Go down a little bit here. This has taken us through lesson 5 up to this point. What you'll be doing as a project in this chapter is you'll be looking at some biotechnologies. We've got some goodies here as far as recombinant DNA, making new sort of genetic DNA, or looking at genetically modified organisms, working with stem cells. There's some really neat stuff here because we can look at the genetics of that and talk about GMOs, and you'll get a chance to really investigate one of these topics, especially recombinant DNA. It's a really neat one because it's a chimera. It's this new genetic, new series of genes that we make by using sort of synthetic techniques. It's not a result of sex. It's a result of scientists messing around in the lab. But stem cell technology, GMOs, etc., just really neat stuff. But that will be part of your project, which will be posted soon enough. Okay, ladies and gentlemen, thanks for listening. This has been another Bleecker lesson, and I hope you enjoyed. Remember, go out, enjoy yourself, relax. It's not all about school. Ciao.